Well, first, I want to say hello to all the listeners in Argentina and other countries. I'm very excited to be here today to share the results from the Paradigm trial, which is a trial of a drug called PRIMEC. I'm working closely with the company Neurosense to develop the next phase 3 trial and to bring this forward for people with ALS all over the world. So today, I'm just first going to tell you my disclosures. I do have an institutional agreement with Neurosense, the company that owns PRIMEC, to help them develop the phase 3 trial. So we're in this amazing state in ALS right now where we have better understanding of the disease biology. So our drugs are getting more targeted. And we do have some treatments available for people living with ALS. There's riluzole, there's Radicava. Those two drugs are for every form of ALS. And then there's a gene therapy, Qalsody, for people who carry mutations in SOD1. So we have made definite progress, and we know that there's ways to support people, improve their quality of life. And there's really a global engaged patient population who wants to be part of research and part of the solution, and global networks to try to help bring trials to people where they live. And there's so many companies working in this space that it gives me a lot of hope that we're going to have some real breakthroughs, and Neurosense is one of them. So let me tell you a little bit about PRIMEC. So PRIMEC is a novel formulation of two FDA-approved drugs, celecoxib and ciprofloxacin. But the combination targets different biologies. And we know that ALS is a complex disorder where there's many different pathways involved. So this is, in a way, a combination treatment. The celecoxib does a couple things. One, it does block inflammation. It's an anti-inflammatory, non-steroidal drug, but it also increases the levels in the brain of ciprofloxacin. So it really has a dual role here. We also know that ciprofloxacin, which is an antibiotic, but it's really being used here in a different way. It's an iron chelator. And we know that in people with ALS, there's something called ferroptosis, which is oxidative damage from iron abnormalities in iron metabolism. And then even more importantly, perhaps, ciprofloxacin works on something called microRNA dysregulation, which is something that we see in all people with ALS and was one of the primary drivers for picking this drug to bring forward. So I'm going to go through some of the preclinical models before showing the results of the recent trial. So there are zebrafish models of ALS that carry no mutations. So there's one with a mutation SOD1 and one with TDP43. And these fish swim slower, less distance, and they have less synapses. And it's a good way to screen drugs quickly. So in the SOD1 zebrafish model and the TDP43 zebrafish models, each drug alone was beneficial in a small amount, but the combination, which you see here in blue, in light blue, almost had the fish swimming the same distance as the wild type. So that was true both in both models. And then when we looked at the synapses, again, the combination of Cipro and Celebrex PRIMEC restored the number of intact synapses in that model. So that was some of the initial preclinical data in a zebrafish model. And there's also data in a cell-based model. And these are iPS-derived motor neurons from people living with ALS. So these are motor neurons that in the hands of Dr. Justin Ishida at USC, they don't live as long as motor neurons derived from people without ALS. And when they treat these cell lines with Cipro and Celebrex, they do improve survival a little bit, but the combination does much more, shown here again in light blue, than either drug alone. But usually we like to see the drugs have a biology in multiple models of the ALS before bringing them to people, and they really do, both in animal models as well as cell-based models. So there was an initial phase 2A trial. This was a small study, 15 people, open label, but this was really to look at safety and biomarkers. And what they learned in this open label study was that PRIMEC was able to decrease the TDP-43 exosome. So they were able to measure TDP-43 in secreted exosomes, and that it also had an effect on markers of inflammation. So this was used to kind of make sure they were going with the right dose in the phase 2b trial. So the phase 2b trial is called Paradigm, and this is a completed study. Participants came in at screening and were randomized 2-to-1 to active drug versus placebo. And it's still a relatively small study, as you can see here. 45 people were assigned to the PRIMEC and 23 to placebo for 6 months. And then there was an open label extension that's ongoing still. And this was done mainly in Israel, Canada, and Italy, I believe. So the main outcome was safety and tolerability, as well as biomarkers, but we also looked at functional rating scale, breathing, quality of life measures, and survival. The two groups were well balanced in terms of where they were in their illness, with the ALSFRS really the similar between the two groups. I'll show you first the safety. Big picture, this was a safe treatment, both in the 6-month period as well as an open label extension. The number of treatment-related AEs were similar between the two groups. There were slightly more adverse events in the treated group, and the most common ones were related to GI, mild GI upset, but nothing that really led to people, a large number of people needing to stop the drug. In fact, the discontinuations shown in the last row were very similar between the active and the placebo group. So good safety, good tolerability. Now we're going to show you some of the measures of clinical efficacy. And the treated group is in blue, the placebo in gray here. And you see after 6 months, about a 29 or almost a 30% slowing of the illness in the treated group on this measure of ALS functional rating scale. Now, the p-value isn't statistically significant. It's 0.12, but this, again, was a small phase 2 study. We now also looked at the data from the open label extension. And here we're really comparing people who started the drug 6 months earlier to people who started it 6 months later. And we see that the group that started it later really never catches up to the group that started it early, shown in blue. And here we see, by the end of a year, about a 36% difference between the early treated versus the late treated group on this ALS functional rating scale. Now we looked at the different subcomponents of the ALS functional rating scale because, as we know, ALS affects people differently in different parts of the body. The 4 domains are the respiratory, bulbar, gross motor, and fine motor, and we see in all of them that there's people do better on the active drug than the placebo, but we see, at least in this trial, the most pronounced effect in the bulbar area on speaking, and the questions around speaking and swallowing. So we also looked at several other outcome measures, and I'll just show you this. Fortunately, most people survived the double-blind period. And even the open label, so we don't really have a lot on survival, but we have shown positive effects on vital capacity, on the King's and Mito stage, and on quality of life as well. So just to summarize, in this 2B study, which was a relatively, it was a good sized one, but not a very, not like a 200, 300 patient study. It was a 69 patient study. We saw good safety and tolerability, which was the primary endpoint. We saw slowing of progression as measured by ALS functional rating scale, both at the at the 6-month mark, but also we saw it when we compared people who started the drug early to those who started 6 months later at the 12-month mark. We saw that it had an effect on multiple different domains, but the most pronounced in speaking and swallowing. While I didn't show you all the data here, there was potential that survival might have been better in the group at 12 months, but the number of events is very small. We saw consistent trends that were positive in all the outcome measures. And this is really, to me, this is a drug that's ready to go to phase 3 for a pivotal, longer trial to confirm or see if these results can be confirmed. So now, thank you for listening to me, and I'm happy to answer any questions.
All right, Dr. Sukowicz, that was excellent. Thank you so much for that presentation. It's really exciting, of course, and more and more people are starting to ask for this compound. It's a growing, I would say, it's a growing interest, at least here in Argentina. So that's why we really wanted to talk to you about this. So okay, so the first question I have for you is kind of a classic question. And so, what's next for this PRIME-C trial in terms of future publication of results? And as always, what is the potential for any kind of conditional approval or expanded access potential for this drug?
Those are both really good questions. I think the next steps are, there's a few. One is, the company speaking both to the FDA and the EMA in Europe and other regulatory groups about requirements. I do think they need to do a phase 3 trial. Given that it was only 69 people, and in short, I don't think it meets kind of the accelerated approval pathway, which is, that's was used, for example, for Qalsody, or the, single study approval with Amylyx. So, that's my personal opinion, that they need to move forward to phase 3. And I think that they're planning that hopefully in the first half of 2025. I think that eventually they'll get, more specific about that, once those conversations with the regulatory agencies happened. I hope that concurrent to the phase 3, there can be a companion expanded access protocol in many countries. And I know in the United States, for example, there's federal funding for that, the Act for ALS, but you have to have an ongoing phase 3 trial. And I hope again that the company will do that, not just in the US, but in other countries as well.
Clear. Thank you. And you know, when we have every time we have one of these combination products, there is always the issue about people going to the pharmacy and getting the two different compounds that are approved for a different indication, or even doctors prescribing it and telling their patients, okay, you can try it, just try it. I mean, if you're willing to do that, I can prescribe the two compounds for you, and there's always a pharmacy near your house that can do the homemade combination, right? So what would be your suggestion or your advice for both providers and patients with respect to this?
Yeah, that's a good question. You came up a lot also with the Amylyx drug before it came to the market. I do think that just compounding it isn't the same as the active drug. These companies combine them in a certain way and work on the PK and the formulation so to optimize its absorption. So that they're really not identical. They're probably close, but not identical. And at this time, I'm kind of recommending to my patients to try to, if they can, hopefully the trial will be soon, is to wait for the trial. I think we really need like a replication of this. It's very exciting data. It's got a good mechanism, but we don't know that it works, honestly, because we've seen other phase 2 studies not repeat in phase 3. We really need this bigger study. I think they meet all the criteria for doing another study. I'm not sure they meet the criteria for rapid, prescribing it to everybody. But I get that it's such a serious illness and people don't want to wait for those things. But hopefully, if the trial can start soon and be global, then people can get access to the real thing that way.
Okay, thank you for that. So we've been having a couple of setbacks lately, right, in the ALS research landscape. Although we had moved forward a lot, and we had made extraordinary progress, I would say, compared to the situation that we had, like, I don't know, 7 or 8 years ago. But in the last few months, maybe we had a couple of setbacks. But what are the learnings that we can take from that?
Yeah, I think that's true. I kind of view it like we have two steps forward, one back, but we're in general still moving forward. But we've had, I think the biggest one really being the repeat Relyvrio study not being positive. I think the Edaravone is more complicated because it was really a different formulation, a different company. I think there are a few learnings. One is that we do need replication in clinical trials, especially first when the first study is small, because the disease is so different between people that you can, by chance get, some differences in the group. So we really do need that. I also think that when we have a positive study, we should try to repeat it as close as possible to the original study. One of my concerns with the Relyvrio repeat study is that the population was different. It was people with the illness for longer, so more heterogeneity in the population. And the trial was much longer. It was 12 months versus 6, with a very high dropout rate, something on the order of 40%. And so instead of a very tight, trial in kind of a homogeneous population for 6 months where they had good retention, the trial changed. So the question is it really that the drug doesn't work, or is that the way you tested it was different? And I don't think we'll ever know because no one's going to do a third study of Relyvrio. So that's one of the lessons I take, is that the next time we have a positive phase 2, like PRIME-C, maybe we should try not to change too many things. We have to change the number, right? We have to go to bigger studies, but try to keep the same inclusion criteria, as much of it the same, so that we really can compare the two trials.
And there's the thing about the time, the duration of the trial, right? People sometimes tend to think, let's do this as fast as possible. Let's try to do shorter trials. But then when you have some cases like what we had with the person, it looks like sometimes if you wait a little longer, or if you extend the trial a little longer and wait for the drug to do what it has to do, well, it might be better. It might bring more positive results. So I guess we are trying to find that balance, don't we?
Yeah,
Are
I
we?
think, yes, absolutely. I think some drugs will take longer to get into steady state and work, and other ones might work faster. But if you don't know about that, you're better off doing a little longer. But it's a balance, because if you go too long, people do, for a variety of reasons, stop participating, and then you're dealing with a lot of missing data. So one of the things that we're doing in our Healy platform trial, which was a 6-month study, we've decided for the next 4 drugs, we're going to 9 months. So it's kind of a compromise that'll allow drugs that need a little more time to have efficacy, but it's not asking people to be on a placebo for a year or more. So it's a compromise. Yeah.
Yes, it looks that
I
way.
do. Yeah. I do hope for the field, though, that we develop short-term markers. Like maybe neurofilament will be the one, or other ones, because it'll just get faster drug development. We're just not quite there yet.
That was actually going to be my next question about biomarkers. We've been really strongly looking for biomarkers. I mean, it's like this disease desperately needs more by efficient biomarkers. Neurofilaments have been around for a while already, but it looks like they are now having a more solid base in terms of being the new biomarker that we're going to be using in many trials. So how's the research doing in terms of that? How's the research working now with neurofilaments, and how's the studies in order to find other biomarkers?
I think neurofilament is quite good, but it might not be the perfect one yet. So we know that especially with a gene therapy that's right on the cause of the illness, obviously there was a rapid decrease of it that predicted clinical response. That's the causality. In a drug maybe like Prime C or one that, works not on the, first cause of the illness, it might take longer and the change might be less. We did see some neurofilament changes in the paradigm trial. And then we know that it's quite variable within a person, so that we think actually that you need to see an effect more than 20% just to get above the noise. So it's good, but there's a lot of learning, and every trial is now including it. But my guess is that in a couple of years we'll be using different things. So some of the new things are coming out that are more in the spinal fluid, biomarkers of TDP-43 biology, things like cryptic exons that coming from Phil Wong and Michael Ward's labs. I think those are going to be really good, especially as we get some new drugs coming to trials that really target TDP-43 mislocalization and misfolding. But we have a lot more to do. But I'm excited. I mean, there's a lot of biobanks, a lot of collection, a lot of people, donating their blood and their spinal fluid, and that's how we're going to develop new biomarkers.
Absolutely, thank you. And in Argentina and in many other countries from the what they call the global south and other regions are really eager to participate in clinical trials. We had a couple here of international clinical trials in Argentina. We had masitinib and we had another one, but people really wants to participate, of course. And I think that brings a different pedigree, right? A different genetic makeups then can make international clinical trials more diverse. But it's also from the logistical point of view challenging. So what would you say is the current approach in terms of how international clinical trials should be?
Yeah, I think first of all, I agree with you. I think they're very important for a lot of reasons. One, to give access to people all over the world to trials and to understand the diversity. I do think that they can add heterogeneity to trials, so that we right now I kind of see the phase 1, phase 2 are in like maybe 1 or 2 countries, kind of the initial proof of concept. And then, one of those countries could be Argentina or Brazil, or a different country. But to try to stay focused on that initial trial, get that, does it work or not? And then when you go bigger, be global, but make sure that you have enough people in each country so that you can look at country specific or population specific effects. But I'd like to see there's the 300 companies they can to get patients, you can go in every country that has a trial network and do your trials. We don't all have to do them in the same country.
Yeah, absolutely, and we're pushing for that here. Yeah, hopefully we will have more trials in South America
Yeah.
soon enough. And what's next in terms of other research projects? We know that you are involved in many. In many, we know how busy you are. So any of if there's any of those projects that you will particularly like to highlight?
Yeah, a couple. So for our platform trial, the Healy ALS platform trial, we're working with 4 companies now to launch in early 2025 the next regimens. One of them we've publicly shared, PharmOst, which has a drug that works on autophagy or the cell death pathway. They've done their phase 1 in Australia, and now they're going to do this phase 2 with us. There are a couple companies that have really exciting approaches for TDP-43, just to keep an eye out on. One of them is Vectory. This is public. They have a way to use gene therapy to deliver an antibody into the motor neurons to clear TDP-43 aggregates. There's a couple companies working on PIC5 inhibitors, also kind of targeting TDP-43, like Verge Genomics, AI Therapeutics. So I think we haven't yet had a study that really looks at the main protein that's aggregated in our illness. It might be that you look at what's the obvious thing and that's not the way to fix it, but it also might be that that's where we should be looking. So I'm excited about those approaches and the development of biomarkers to be able to measure TDP-43 biology in people. So those are a few things, but there's so much going on that, that you can have a talk on each one of those topics.
Fortunately, there's so much going on. Indeed, yes, yes,
Yeah.
it looks exciting. Yeah, absolutely. Okay, so thank you so much, Dr. Sukovich, for that. That was an excellent presentation. Thanks for having a talk with us. Once again, this is not the first time, so we really appreciate that you are sharing some time of your busy schedule with us. So hopefully
Thank
we
you
will
for
see
having me.
you very soon, maybe in Montreal, and maybe in Argentina as well.
Yes. Yes.
So we are looking forward to hearing more news, exciting news about your projects, and to seeing you very soon again.
Yeah. Thank you for having me. Have a great day.
You too. Thank you so much, Dr. Sukovich. Take care. Bye, Mary. Thank you.
Bye. Thanks.
Have a nice day. Thank you.